Have you ever taken antibiotics? If yes, then did you know that some genes can evolve to be resistant to the antibiotics you take? Although they can protect us from harmful microorganisms like bad bacteria, antibiotics also create a selective pressure. Due to differences in their DNA, some bacteria can survive. The genes that promote that survival are known as antibiotic-resistant genes, or ARGs. A group of researchers in China investigated these ARGs in the context of estuaries. Estuaries occur whenever rivers meet the sea and fresh and salt water mix. This type of environment can naturally filter out pollutants, but poor water quality, which is caused by pollution, impacts many of the organisms that live in estuaries. This makes estuaries an easy place to see how anthropogenic, which means human, activities impact the environment. These researchers looked at factors like bacterial composition, environmental factors, geographic locations, pharmaceutical residues, and human activities. They hypothesized that human activity would be the strongest driver. First of all, researchers took samples from 18 estuaries over 4,000 kilometers of coastline in China for a total of 90 sediment samples. They used PCR to investigate the spatial distribution of ARGs and statistical tests including partial redundancy analysis. Here are some of the more unfamiliar terms. ARGs, or antibiotic-resistant genes, are genes that protect a microorganism from antibiotics and resist them from being killed. To characterize the different bacteria, the 16S rRNA gene was extracted through the samples. The 16S rRNA gene is commonly used in characterizing bacteria as it undergoes evolution at an extremely slow rate. Essentially, a bacterium species is expected to always have the same 16S rRNA copy number, regardless of whatever other types of changes occur, for instance, building a resistance to antibiotics. By extracting ARGs and knowing the relative bacteria, it is possible to further analyze the factor of bacteria type in the formation and pollution of ARGs. Composition of bacterial communities were obtained with the 16S rRNA gene sequences. Overall, the 18 estuaries were similar at the phylum level with three main phyla present: Proteobacteria, Bacteroidetes, and Chloroflexi. Remember this sort of dot diagram in Module 2 when we were looking at species richness and evenness? If researchers came back and did this analysis over several years, they could probably collect evidence of evolution or speciation via the Shannon Diversity Index. Some of the major findings included that when streams contained residual antibiotics, the corresponding ARGs were often present. We talked about this in our evolution module. This makes sense and indicates evolution because ARGs can be present in a population simply from random genetic mutations. When these antibiotics enter the water system and become selective pressures, less resistant bacteria are killed and the bacteria with resistant alleles survive to reproduce. This process continues as more and different antibiotics pollute the waters. To see exactly how bacterial composition, environmental factors, geographic locations, pharmaceutical residues, and human activities were related, a partial redundancy analysis was used. This test looks at redundancies or overlap in factors and shows 80% of the observable variations in ARGs could be explained by a single factor or combination of factors chosen, leaving less than 20% unexplained. Anthropogenic factors appeared to be the individual major driver behind the variance at 27.22%. Since the sampling was done along the 4,000-kilometer coastline and no spatial pattern was found, it is also unlikely that environmental factors like climate were behind the diversity and abundance of ARGs. Bacterial communities, which were hypothesized to be a key contributor to variance in ARGs, were shown with the PRDA to play a relatively small role, 2.61%. Overall, Chinese overusage of antibiotics allows bacteria to build up a resistance to treatment, therefore reducing the overall effectiveness over time. This study also shows how widespread the pollution from antibiotics and corresponding rise in ARGs has become. Due to human actions in compilation with environmental factors, ARGs pose a severe threat to future health of both the natural environment and its inhabitants.